Great. Thank you, Tome, for this very nice introduction. Let's see if this works. It does. Okay, great. It's great to be here. Today, I will talk about large-scale prediction of phenotypes from biological networks. And this might be more tangibly related to nutrition, but I think it might be interesting to talk about the kind of methods and problems that we are tackling, and hopefully I will talk about ideas that might give you or be useful for your research as well. So when I talk about biological networks or networks in general, what I have in mind are these complex systems that typically consist of very interconnected objects that interact with each other in some way. So the way to think about that is that we have a system where we have a number of objects, say those red points, and those red points are connected in some way. They depend, they interact with each other, they depend on each other to achieve a particular goal, and that is something that we think of as a network or a graph, obviously. So why networks and why graphs are the central point of my research? Primarily because networks are a very general mathematical language that allow us to describe very different complex systems. So we can use the same mathematical machinery to describe, for example, relationships between actors in the sense of who appears in the same movie with whom, and then also about, we can use these networks to represent friendship relationships, or in a completely different domain, how proteins, molecules, and human body interact with each other. Underlying these three very different examples is the same mathematical object, which in our case would be this very simple toy graph on 4 nodes and 4 edges. So it turns out that in biology, medicine, and health in general, there are many, many data sets that actually come in the form of graphs and networks. In particular, that might be the case because in all those domains, it is the objects of interest typically do not act independently of the environment, but are somewhat embedded in a larger context that might be a cell, that might be a tissue, a human body, or society, and within that context, those entities interact with each other. For example, that might be in the context of interactions between patients within a society, to interactions between cells in human tissues, interactions between genes, or just to think about how we can represent knowledge of biology, medicine, and science in the form of large knowledge graphs. So this is great, and many data sets are now networks, but now with the question that we might have in the current era of learning over large data sets is, how do we effectively learn deep models on such complex systems such as biomedical networks? And what I have in mind here, ultimately what our goal is, we want to have a methodology and approach that will start on the left with a particular representation of our problem in the form of a large network, and then it will be able to learn and reason over it over a series of potentially nonlinear transformations to reach to obtain predictions at the end that might be predictions that tell us something about patient outcomes, that are useful for discovering new disease genes, identifying unwanted side effects about drugs, predicting drug treatment response, and so on. So networks are powerful data representation. We have seen that in the previous few slides. However, it's quite challenging to work with networks for prevailing machine learning models. And I will elaborate on that in the next, in the next slides and in this talk. So in the first part of the talk, I will describe the methodology that is known as network embeddings or graph neural networks more generally, that really allow us to train and learn those deep models on large graphs and of different biomedical entities. And then I will show how that methodology has helped us tackle 2 very important problems. The first that I will focus will be that of predicting safety and side effects of combinations of drugs. And I will give concrete examples how the methodology that we developed is useful for that problem and how it has led to exciting follow-up research in actual clinic and biomedical research. And then I will also spend a few minutes to talk about about how we can use those methods to gain insights into human diseases, where in particularly I will focus on how important it is that we are very careful when we are modeling data that comes from different domains. For example, social networks versus biological networks. Okay, let's start with the first part of the talk. And here, really, without being too technical, I want to give a gist of what is at the core of the methodology that allows us to tackle those important problems. So remember, our goal here will be how can we learn those deep models and deep representation learning models on really networks or graphs as inputs. And as I said, that networks are challenging for prevailing machine learning models. What I mean by that? What I mean by that is that prevailing deep models are primarily designed for grids or simple sequences. So many of you are familiar probably with convolutional neural networks and many of their variants, which are primarily designed for fixed size images or grids, where you can think of every pixel being a point or a node in our network, and that pixel has exactly 4 neighbors, which is pixel to the top, bottom, left, and right. On the other hand, a very important class of deep learning models, known as recurrent neural networks and all of their variants, are primarily designed for text or sequence data. And again, you can think of those data sets as being very simple graphs that are pathways where every token, a word, or perhaps a nucleotide in biological sequence would be one node, and then nodes are connected based on the sequence at which they, based on the order in which they appear in a sequence. So these models have really brought extraordinary gains in many fields of computer science, including computer vision, natural language processing, speech, and robotics. However, they are really not able to consider interactions, which is the essence of real-world networks. What I mean by that is that real-world networks do not look like grids. They do not look like linear pathways. They look like objects like the one shown on the left, where different nodes have different number of neighbors, we have interesting local and global structure, and they might be even more complicated. In many real-world settings, we have different kinds of objects that interact with each other, so we have those multiscale, multimodal networks. So deep learning on networks is really hard because biomedical networks or networks in general are far more complex. Technically, the kind of structure or problems that we would have if we would naively apply those classical other deep models to network data is that at the core, in networks, there is no spatial locality, meaning that for a particular node that we consider, there is no fixed ordering of the nodes of the neighbors that this node interact with. That's in networks. However, in the context of other grids or sequence-based data, there is a fixed ordering, and we can talk about pixels or tokens that appear on the left and right, top and bottom. But in the context of networks on the left, this is something we cannot discuss. There is no necessarily in society, there is no a human to the left of you. It's just like, it depends on who you interact with. And those people might be very far physically, so there is no notion of being left or right or up and down. Okay, so in the next few minutes, what I will overview will be the methodology that allow us to very flexibly learn those deep models on actually network data, where you can think of it at the high level as being an approach that will start with a multimodal network on the left, which I will define in the next slide, and through a series of nonlinear transformations, we will reach predictions on the right, which are then useful for a particular problem. So the setup here is that we operate with that idea that our input consists of a multimodal network. Multimodal network is a network where we have data that describes different kinds of entities that interact with each other in different ways. Here's an example of a multimodal network where we have mode number 1. It might be, for example, drugs. So every green triangle would represent one drug, and an edge between two drugs could represent a specific kind of interaction between those two drugs. Of course, we might not have only information about this specific mode of drugs. We might have information about how those drugs actually operate and affect human body. So we would know what are the interactions between drugs to proteins. And proteins are now the second mode of information, because these are entities that are conceptually, of course, very different from drugs. And of course, proteins can interact with each other, which gives us connections or edges within the protein mode. So altogether, what we have here, or what I'm showing here, is a multimodal network that is quite simple. It has just two modes, and then which are shown here in the form of green triangles in orange circles, and then we have edges within the mode and between the mode. So assuming that this is and this will be the working example for us today, this network. So assuming that we start with a network, the high level, our approach has two components, if we want to phrase it in this autoencoder perspective. And the first component is the idea of encoder, where what it does, it takes our network that we have seen in the previous slide, it learns an embedding for every node in that network, and I will define embedding next. And then in the second stage, what we do is when we have the embeddings, when we have learned what is the best way to project our network to some low-dimensional embedding space, which is compact, then we leverage that compact space in order to make predictions. So just let me spend a minute on the encoder component. The notion here is, or the key technical idea is that idea of embedding nodes in the network. So what I mean by that is that for every node in the network, say node U and V, what we learn is some mapping function f that what it will do, it will map every node to some d-dimensional feature vector or latent representation of that node, which is called embedding. Now, the whole idea, or the crust, is actually, or complexity is hidden in that function f. And that function f is defined through a notion known as graph convolutions, that what it does, it operationalizes that idea that two nodes that have similar neighborhoods on the left, so that have similar local interaction structure, should be embedded close together in the embedding space. So the idea is then to learn mapping function f such that the structure of the embedding space would reflect topology of the input graph. And that is really important because for prevailing machine learning models, it's very difficult to work with graphs and networks because of these combinatorial structures, but they're very well used to working with large feature tables. And by learning those embedding in effective ways, we get feature tables that effectively capture topology of those graphs that we have as input. So really the goal of mapping functions f is that we want to map nodes to some d-dimensional embeddings, representations, or vectors of those nodes, shown here as those blue rows or vectors, such that nodes that have similar network neighborhoods will be embedded close together. Okay. So that is the core of the encoder component. Without going in details, when we learn such mapping function f that can generate embedding for every node in the network, then we use those embeddings that we have learned to make predictions. So what kind of predictions can we actually make? So there are different kinds of predictions we can make. I'm just highlighting here four conceptually very different types of predictions. So what we can do is we can predict a property for every node in the network, and that is known in network science machine learning language as node classification. Or we can predict whether two nodes are associated with each other. That would be known as link prediction, for example, if you want to discover new friendship relationships, or in the form of node classification, you want to predict what are functions of certain genes or proteins in human body. We can use those learned embeddings and simply cluster them to detect groups of entities that show similar behavior, or we can learn embeddings not for every node, but we learn them for subgraphs or graphs, and then use that for new questions, for example, in chemistry. But really, to recap at the high level, what this means that with the development of graph neural networks and network embeddings, we can apply deep learning much more broadly, not only to medical images, not only to DNA sequence data, which really opened doors to many new kinds of problems that we can now tackle, and my focus is specifically to problems in medicine and biology. So I didn't go into details about the technical parts, but we have a tutorial that with around 300, 400 slides that really goes from the introduction to state-of-the-art models, including all the mathematical derivations, including code bases, IPython notebooks with examples and everything. So those that are interested in learning more about the methodology are welcome to check that website. Okay, so let me now, in the rest of the time, describe those two important problems that we tackled. And the first one will be how to use network embeddings, this idea of learning low-dimensional representations for complex multimodal networks in order to model combinations of drugs. So to first motivate this project, a good question that you might ask yourself if you don't work in this area is, why would I even care about combinations of drugs? So this problem is known as problem of polypharmacy, and it actually most often refers to the situation when patient takes multiple drugs at the same time. So they take a combination of drugs. Why do they do that? To treat complex or disease or many coexisting conditions. So a bit of statistics, which is statistics for the US. 46% of people of 65 years or older take more than 5 drugs at the same time. It's not uncommon to encounter patients that take up to 20 drugs to treat very complex diseases, such as heart disorders, depression, cancer. And that is okay. The problem, however, becomes that with a large number of drugs that are being taken by individuals, this increases the likelihood of unwanted side effects, which is a burden for the healthcare system as well as for individual. So what do I mean by unwanted side effects? What I mean by that, and I will illustrate this with the following example of a table that sits actually at FDA, so that is the Food and Drug Administration in the US. And this table is known as the FDA Adverse Event Reporting System. It's a national database where for every patient in the US, for every individual that takes prescription drugs, that individual gets at the doctor collects information about potential side effects that individual experiences, and that is recorded in that national databases. So imagine we have this nice table here, and it has 2 columns. So we see what are the drugs that are associated with what kinds of side effects. So we can read that as 5% of patients that take this red pill experience or develop hepatitis as a site of unwanted side effect. So what I wanted to bring your attention in this slide is to the following situation. So let's look at the red drug, and we see that the red drug is associated with 3 kinds of unwanted side effects. And similarly, green drug is associated with 3 different kinds of side effects. For patients that take green and red drug together, the kind of side effects that they experience is exactly what you would expect to see. Those are side effects that are experienced by people who take either drug alone, right? However, if you look at another example, which is green and orange drug, we see that patients that take green and orange drug together, they experience side effects, and they might develop colon cancer at a certain rate and experience fatigue, which is something we do not see in patients that take either green drug alone or either orange drug alone. And that is unexpected. We would not expect that to see if the 2 drugs, orange and green drug, would not somehow interact with each other in a way that we do not want. So this is known as an unexpected drug-drug interactions. What it means, a bit more formally here, it means the following. So imagine we have a system with these 2 pills. We have a blue and red pill. When a patient takes blue pill, everything is okay, no side effects. When a patient takes red pill, everything's okay, no side effects. When a patient takes both pills together, the patient will experience certain side effects. And that is something we would not expect because it's not due to linear assumption about the model. Our task here will thus be to have a model that can predict how likely a particular combination of drugs will lead to a particular type of side effect. Why is modeling this particular problem very challenging? So it's challenging for a number of reasons, but let me mention three of those. The first being a really combinatorial explosion of possible combinations of drugs. Just if we would consider all combinations that involve just 2 drugs on the US market, we would get more than 30 million possible combinations. If you look at combinations of 3 drugs, we get more than 20 billion possible combinations. Because of this combinatorial explosion, combinations of drugs are basically not studied in laboratories, through actual experiments during the process of drug development in pharma. But we almost are doing those natural experiments when patients in real world, when the drugs are approved, take combinations of drugs and we see what happens, which is not probably what we want. So also drug-drug interactions are non-linear and non-additive. Exactly what we want to measure is to want to capture something that's a deviation from the expected behavior, which would be linear additive model. And you can think of that as a big data problem, because what we will work on with information about all patients in the US. So it's a big data set. However, it very quickly becomes a small data set problem because there are so many possible combinations of drugs, and for a particular combination, there's only a small number of patients that take that combination of drugs, and even fewer percentage of those patients experience side effect. So that means that the kind of models that we would develop that would operate on a per drug combination or per side effect basis that would be independent from each other are simply not possible, because there's no way we can train the models using only that information. But we need to have one joint model that can capture and learn over the entire information and capture potential similarity and interdependence between side effects. So in order to tackle this problem, the first thing we need to do is we need to build really high quality data set that allow us to operationalize this problem and work with it computationally. So our objective was to capture molecular drug patient data for all drugs that are prescribed in the US. And for that, we teamed up with the FDA and we built a very unique multimodal network, which is actually the network that I used before as a working example network when describing embeddings. And it's a network that has these 2 modes of drugs in green triangles and proteins in orange circles. And the way you can interpret this network is, for example, an edge of type R1 between 2 drug nodes, C and M, indicates that when patients take drugs C and M together, they experience side effect R1, which might be gastrointestinal bleed side effect, and that side effect cannot be attributed to either drug alone, right? And then, of course, between any pair of nodes, any pair of drug nodes, there might be many, many different kinds of edges that would indicate different types of side effects. And those edges are, of course, weighted because they're different, because there's certain likelihood associated with the patient developing the side effect. And in order to be really able to understand our predictions, we really need to know how drugs operate and how drugs affect human body. And that's why we have the second mode that tells us about what are the proteins or the molecules in human body that are actually affected when a patient takes a particular drug. And those are proteins that are targeted by the drug. Okay, so the embedding-based approach that we developed, that I highlighted earlier in the talk, we applied it to that polypharmacy data set and network, and that allowed domain experts to ask questions and pose questions to the model. So, for example, a user of the system can ask now questions such as how likely it is that a form of statin drug and ciprofloxacin, when taken together, would lead to breakdown of muscle tissue. So that is a query a biomedical researcher or a chemist might ask, but formally what that means is we are asking the underlying ML model to predict how likely it is that an edge of type breakdown of muscle tissue would exist between the 2 drug nodes C and S that represents the drug nodes that that were in the query of the user. So we trained our model on the large data set, and the first thing we typically do as we always do as developers of this kind of methodology is we evaluate approach in a series of empirical experiments using cross-validation setting, where we put certain data set, a certain part of the data aside, and then we train our model on the remaining data set, and then use the model to make predictions, which we then validate against the test data set. So we did that in order to understand if all components of our approach are really necessary and positively contribute to the performance, and if it is embeddings that are useful and crucial and really important for accurate prediction. So it turns out that those questions were actually confirmed, and what I'm showing here is on the y-axis there is the AUC score, which indicates performance accuracy. And higher is better. You can interpret is the probability that we will actually recognize the correct side effect associated with combination of drugs. And then comparison with the different simplified variants of our methodology show that all these components and embedding-based approach is really important. Beyond cross-validation, what we wanted to do is really to see, are our predictions useful in real world? So the first test that we did for that is we took our approach and we trained it on all the data generated prior to 2012. And then we asked the model, well, if you have seen all the data up to 2012, what are the predictions that you make? And then we ask, when we looked at those most top-ranked predictions made by the model and evaluate them against the data that was and findings that were published and discovered after 2012. So we kind of almost use this time machine back approach in order to pretend as if today is 2012 and how well does the method perform. So what I'm showing here in this table are top 10 predictions made by the model. So each prediction, as we have seen before in an example, it's a prediction that connects two drugs with a particular side effect. So for 5 out of those 10 top predictions, we were able to find direct evidence after 2012 that confirms our prediction in the sense that there were concrete groups of patients that were on specific combination of drugs and have actually in practice experienced side effects that our model predicted. And that has really been fantastic and has motivated some follow-up research. But then we also ask, well, what about those 5 other empty spaces? Those might be simple mistakes by the model, or they might be new hypotheses that actually are worth looking into. So in order to validate those new predictions beyond those time machine experiment, we are now have established partnerships with a few hospitals at Harvard and Stanford, and we are now clinically validating those predictions in several clinics where our AI system is actually sits next to a physician, and physicians can do their work the way they do it every day, but they can also consult our approach to make recommendations, to rank those recommendations, and then consider those recommendations when making treatments. And we are validating several predictions that way, and so far it has led to very interesting follow-ups. So one of them is to develop personalized treatments in the sense that we can think about what are combinations of drugs that are the best for specific patient that might come from a specific environment, have a certain age, and have certain preferences and lifestyle. And can also this approach be used to motivate new design of new combinatorial drug therapies, for which we have a follow-up on prostate cancer and schizophrenia at Stanford. So this is this first example of how really methodology that we developed and the core methodology published in, I would say, very core machine learning venues can be fairly quickly translated to an impactful real-world case where we can show how the methodology can affect and really help humans. Now, the second part will be more about how we can gain insight into human disease by leveraging and studying biological networks. So this is really important question because in order to be able to treat the disease, we first need to know and learn something about how disease actually affects human body and what potentially the causal mechanisms that would cause the disease or that are associated with disease in order to be able to treat it. And we want to do that computationally and be able to automatically extract interesting insights. So if you don't work in this area, which I guess many do not, you can think of that as a problem where what we care about are, we want to have tools that will allow us to answer questions such as, what does my protein do? So imagine we have that biomedical researcher who might be user of that systems and they're very excited about their own favorite gene or protein, and they want to know what does their protein, what their protein does, what are diseases that the protein is associated with. So at the high level, the way you can think of this area of research, it uses the following pipeline. So it's all about integrating very large different kinds of information about proteins that might be genomic data about functions that proteins are involved in, and taking known labeled information about proteins and diseases that we already know about. And then our protein prediction methods are those methods that can then answer the questions about what certain protein does in the form of producing those ranked lists that would say that a protein AXIN1 is the first most likely protein to be associated with a certain disease that this example would care about. So as an example, instead of thinking about disease in a very abstract way, example might be Meckel-Gruber syndrome, which is a rare a recessive disorder caused by dysfunction of cilia. And for that disease, there are a few proteins that are already known to be associated with that disease. Those three on the left. What we want to do is we want to find what are other proteins that are involved in the onset of that important and unfortunately quite horrible syndrome. Okay, so let me talk a bit about notation here, and terminology. When I will use the term disease-protein association, that means that I refer to the situation where there is an established association or a link between a disease and a protein. That the way we'll think about it in this talk is that some form of malfunction or alteration in that protein, in that molecule, is associated with the onset of a disease. And if we collect together all proteins the disease is associated with, what we get is the disease pathway. This is the set of proteins associated with the disease. And those proteins do not work independently of each other, but really the way those proteins work, they are embedded in some underlying biological network, which is known as protein interactome. So this is a network where every node is now protein. There's an edge between two proteins indicating physical protein-protein interaction, meaning that proteins as molecules are physically close in the cell in order to achieve a particular goal. Now, certain proteins are now disease proteins. So, for example, in this toy example, we have those 5 proteins, 3 are labeled red, which would indicate that we know that for 3 out of those 5 proteins, they're associated with a particular disease. And if we know just 3 of them, that would mean that the current disease pathway consists of those 3 proteins and their interactions with each other. Now, it's a good question if MOB4 is also a disease protein, given that it interacts with another disease protein. That's something we want to answer. So this is just an example that we have seen before, Meckel-Gruber syndrome. This is how the pathway for that syndrome looks like. And you can see that there are edges in interactions actually between disease proteins. And in fact, it turns out that that proteins involved with the same disease tend to interact with each other. They tend to cluster in the same network neighborhood. And that is really a principle and hypothesis that is known as disease module hypothesis, meaning that what we expect is the disease proteins will form clusters in networks. That is similarly as we expect, for example, in friendship networks on Facebook, that people with the same interests will form clusters or groups, or people that went to the same high school will also form groups in friendships networks. And there are many diseases where we actually see that this is the case. For example, for mitochondria complex 1 deficiency, this syndrome, we see these are the disease proteins for that disease. And you see that they are very tightly interconnected with each other. It's almost a click. And actually, if we do statistical tests, we see that they are substantially more strongly connected than what we would expect by some random chance that those disease proteins would be. It's very unlikely that just by random chance those disease proteins would be so tightly coupled to each other. Okay, so what it means now is then if our goal is to answer those questions and help biomedical researchers to find more proteins like this, like to start with a known set of disease proteins, and given that we know about disease module hypothesis that says that proteins that have the same phenotype cluster in the network, then it's obvious what we want to do. We want to look locally around known disease proteins and say that, well, those proteins that are very close, that are immediate neighbors of known proteins are likely to be candidate predicted disease proteins. And this has been a very long-standing paradigm in principle that has led to numerous methods and really a number of successes in research over the past 2 decades, including lots of translations for computational prediction to experiments. And this long-standing paradigm really is only based on this notion of disease module hypothesis. Nodes with the same phenotype, nodes with the same property, which proteins with the same phenotype cluster together. Well, that is great, but now the however comes. So it turns out that with more and more data being available and those high throughput experiments being done in biology that can really generate high quality data sets, it turns out that disease module hypothesis actually fails for most human diseases. So most diseases actually do not look like the one that I showed you before, the clique of disease proteins that just tightly connect with each other. They look like that. You almost see that out of those red disease proteins, there's just one interaction that connects to proteins. All others are not really connected to known disease proteins. Another example is like we have many disease proteins, just 3 edges. And for example, there is one protein who's quite far away from, it's not connected to any other disease proteins. So what we figured out is that proteins are actually more likely to have similar phenotypes, so have similar neighbors, not if they directly interact with each other, but if they have mutual common neighbors. So this is the same disease I showed in the previous slide. You see there are no edges between disease proteins. But if we show common neighbors, which are those blue nodes here, all of a sudden what we get is disease pathway. So what this now means, or fundamentally what it shows, is the following. So most of the methodology for disease protein prediction and lots of methodology actually for biological networks actually comes from algorithms that were initially developed for other types of domains. Most often those are social information networks. And the core principle in social information networks is actually the friendship principle, also known as homophily, or translated in biology, disease module hypothesis. What it does, it says that you're more or less what your neighbors are. So imagine we have this small network of friendships where nodes would be individuals, and we want to predict what is the activity that white guy would like, and we know that 4 out of 4 of its neighbors enjoy playing tennis. So we predict that, of course, he will also enjoy playing tennis because his friends enjoy tennis. Well, actually, in proteins, it's not like that. If we want to predict what is the function of that gray protein, it's because 4 of its direct neighbors are blue. It does not mean that that protein is blue. It's because its second order neighbor is red that protein will be actually red. There is lots of fundamental biology why that 2 hop neighbors make sense, but this does now mean that we have this new paradigm which we call mutual interactors paradigm. Okay, so the first question to test this more reliably is of course, is that mutual interactor principle really valid in biological data sets and biological networks across a variety of biological networks, from genetic interaction, protein interaction networks, and so on. We have shown that the principle applies much more strongly than disease module hypothesis. And that has then really motivated us to use protein embeddings as a way to operationalize mutual interactors principle, sort of to make it really amenable to computational analysis and to our deep models. So what we have developed is then an approach that is based on embeddings, this idea that I mentioned in the first part of the talk, that what it allows us to do is to identify new disease proteins, but not based on homophily or direct neighbors, but based on the mutual interactor principle, and also based on a number of other principles that one could very simply encode in the form of equations. Without going much into the details, I will just conclude with a few experiments. An experimental setup here to run those experiments is as follows. So what we assume is that we have our underlying protein interactome, so the human protein interaction network, and we have information for many, many diseases, let's say for 2,000 diseases. We have information about, for each of those 2,000 or 1,800 diseases, what are the proteins associated with that disease. We split those associations in a tenfold fashion and then do protein-wise cross-validation, which means that we pretend that there is a new protein that we don't know anything about. What can the model predict about it? And that's really a hard setting because it means that we want to make our methods generalized to a model, to a molecule that the model has not seen any time before. Okay, so what it turns out that if we operationalize mutual interactors principle and use that as a similarity function for our embedding approach, rather than direct neighborhood friendship principle, that immediately leads to 65% improvement in disease protein prediction. So without really making substantial other changes to our embedding methodology, just denoting what it means for two nodes to have the same label, that yields substantial improvement. And that improvement is really improvement across a large number of diseases, of those 1,800 diseases that we have covered. And what I'm showing here is comparison of our approach to some of the classic methods, as well as neural-based neural or graph convolutional networks that do not operate on that mutual interactor principles. And you can think of it on the y-axis, what is shown is just the recall of our approach minus the recall of the competing baseline. So the more red area that you see, the better the method performs. And you can see that for many, many diseases, actually the method performs really well. And the mutual interactors principle is great. So at the high level, the point that I'm trying to make here in this third story of this talk is really that one needs to be very careful when translating algorithms that were initially developed for a certain domain, translating those algorithms for a different domain. In this case, because the friendship principle was developed and noticed decades ago in the context of social information interactions, and then people, without really questioning those assumptions, translated it to biological networks. That has really led to 20 years of work in biological networks that have used the principle that is actually not the most valid principle for biological networks. And I think it's really important to be able to be aware of that and think deeply about the approach coupled with the machine learning methodology that then allows us to realize that. Okay, my final thoughts is that I'm really excited about how those large data sets are transforming science and medicine, and new machine learning methods such as embedding methodology that I mentioned today can really unlock those data sets, can open doors for scientific discoveries. And networks in particular, and coupled with embeddings, allow us to learn high-quality, actionable models over very complex data sets, and they're actionable because they lead immediately to downstream experiments and inform direct experiments in the lab. Thank you so much.